Our first speaker is Luca Pandolfini, who studies environmental sex determination in turtles. He does that at the Italian Institute of Technology. Luca, take the stage, please.
So, hello everyone, and I want to thank the ONT team for giving me the chance to present my data. So today we are going to talk about this little fella, the tiny tortoise Pyxis arachnoides, which is one of the smallest tortoises in the world, which is unfortunately critically endangered by both his low reproductive rate and by increased threat to its natural habitat. This tortoise comes from southwestern coastal regions of Madagascar and from a very important biodiversity hotspot. This is the so-called spiny forest, xerophytic environment, which is threatened by a lot of man-related activities. And therefore, its population has been depleted starting from the 90s to near to extinction rate. Unfortunately, though, we need to take actions in order to preserve this organism. In order to take action with conservation strategies, the determination of sex and parental lineages of a species are of paramount importance. And nanopore devices, being portable, are really a good asset to these kind of conservation campaigns. Tortoises, as most reptiles, are characterized by environmental sex determination. Therefore, is a non-chromosomal sex determination mechanism, following the rule of hot girls and cool guys, in the sense that at warmer temperatures you have a higher chance of getting female and at lower temperature of getting males. But unfortunately, especially in very young individuals, the confirmation of their sex is very traumatic and invasive. On the other hand, reptiles have nucleated blood cells, so you have easy access to the genetic material. So by a combination of 110 and 114 reads, and also by Pore-C, using the latest Kit 14 sequencing kits, we were able to collect from 50 to 100x coverage of the genomes of this organism. And we put in place a pipeline of assembly by using next de novo, next polish, and 3D DNA canonical assembly that allowed us to get chromosome-level reference genomes in less than 4 months. It's really amazing. And by all means, all qualitative parameters of the genome we obtained was in line and comparable with what you can obtain by standard Illumina or PacBio-based reference genomes for other turtle species. And also the synteny was very well in line with already available turtles genomes. And finally, we were able to identify a few hundred of high-confidence promoter-associated differentially methylated regions that constitute a footprint of what distinguishes from an epigenetic point of view a male from a female tortoise. So next step is to adapt this strategy and scale it down to very shallow sequencing devices in order to be portable in the field. I want to thank you, all the collaborators from Aquario, the University of Zurich, and Org1 for giving me the opportunity to carry on this project, and also you for your attention. Thank you.
Yeah. Thank you, Luca.